Hello, good morning, good afternoon, and good evening from your corner of the world to mine. My name is Sadye Paez, and I'm thrilled to be welcoming you to the 5th Annual Biodiversity Genomics Conference. Hola y bienvenidos. Mi nombre es Sadye Paez y estoy orgullosa de estar aquí con todos ustedes en el 5º año de la Conferencia Global de Genómica de la Biodiversidad. I am a Senior Research Associate at the Rockefeller University. I am also a chair of the Justice, Equity, Diversity, and Inclusion Committee of the Earth Biogenome Project. Essentially, we think about how to reflect the global diversity of peoples and their perspectives in participating in and benefiting from the mission of sequencing all life on Earth, which is also why we are gathering as a global community under the Earth Biogenome Project umbrella, striving to be inclusive in all aspects of biodiversity genomics. I would like to acknowledge the traditional, ancestral, and unceded territories on which we are learning, organizing, and working today. I have the distinct honor of officially opening the meeting, and it is in this capacity that I welcome you on behalf of the Scientific Planning Committee, our sponsor, the Tree of Life, and all of the chairs and speakers who have organized incredible sessions to bring forward their research and to share their passions with you. I'd like to introduce you to some of our community members now.
Hey.
Hola, buen día.
Konnichiwa.
Habari.
Hola.
你好。
Dipaye Kenemun.
Terve ja terveisiä Suomesta.
Iri Unaber.
Bonjour. Pozdrav.
Labdien.
Namaskar.
Guten Tag.
Välkommen till Norge. Hej eller god morgon.
My name is Joel Ogwang, and I am a trainer for the African Biogenome Project in Eastern Central Africa.
My name is Henrik Lantz, and I am involved in the European Reference Genome app.
Jaakko Pohjoismäki. I'm professor in molecular biology and genetics here at the University of Eastern Finland.
My name is Adede Hawinyo-Dero, and I'm affiliated with the Africa Biogenome Project.
My name is Marta Riutort. I am a professor at the University of Barcelona, and together with Dr. Del Campo, we co-chair the Catalan initiative for the EBB.
Buongiorno o buonasera e grazie per il vostro interesse nell'Earth Biogenome Project.
Musta. I'm Francis and I work at the Wellcome Center Institute in the United Kingdom, and I am originally from the Philippines.
My name is Martina Pavlik. I live in Croatia and I am employed at Ruđer Bošković Institute in Zagreb as a research associate.
My name is Elena Bujan. I'm from University of Primorska in Slovenia. My hometown is Koper, which is situated close to the Italian border on the beautiful Adriatic Sea and near to the Slovenian Kars region.
Hola, my name is Melissa Hernandez Poveda and I'm a PhD candidate at the University of Melbourne in Australia.
I'm Dr. Abha Deshpande. I'm a marine biologist with specialization in marine ecology and genomics. I'm based in India. I work in academic and industrial research. I also teach whenever I can.
Je m'appelle Solène Correa and I am the scientific director for Wise Ancestors.
Céad míle fáilte agus go raibh míle maigad as teacht chuig cuideáil na bliana seo ar Genome Big Tagsalach 24. Or, in other words, welcome and thank you for joining us at this year's Biodiversity Genomics 24 conference. This year, our Scientific Planning Committee has been working hard to put together an exciting program for you. It is jam-packed with incredible researchers and research from across the globe. We have sessions from Europe, Africa, the Americas, Asia, and Australasia. We have 10 EBP-affiliated project sessions, 8 technical sessions, 2 sponsored presentations, 2 keynote sessions, and we also have dedicated sessions for early career researchers, ethics, legal and social issues, justice, equity, diversity and inclusion, and Indigenous data sovereignty. To polish it all off, we have 2 wonderful musical sessions that are given to us by Vostera. But before we get started, just to remind you to please read the conference's Code of Conduct, which is located under the Legal Information tab on the conference website. Now, with that, again, welcome, and now let's get started.
fantastic week ahead of us. And to get us started at our welcome session, we have two talks today by Harris Lewin and Mark Blaxter. Harris Lewin is the chair of the Earth Biogenome Project, and he is with the Arizona State University. He will be speaking today on the current progress of the Earth Biogenome Project, as well as discussing plans to complete phase 1 and to initiate phase 2. We will then have a talk by Mark Blaxter, who is the lead of the Tree of Life at the Wellcome Sanger Institute. He will be speaking on building biodiversity genomics around the world. I'd like to pass it on to Harris to get us started, and welcome once again.
Thanks very much, Sadie. Appreciate the introduction and the great work of you and Maja and all the other people who produced that wonderful welcome video. This is my first talk, at least live, at 6 a.m. here on the West Coast of the United States, when most men are either sleeping or perhaps even dead. And so please, I hope you'll bear with me this morning as I hope I can synchronize my brain and my mouth. So it is my pleasure as the chair of the Earth Biogenome Project Executive Committee to welcome you all to the Biodiversity Genomics 2024 virtual conference. You are joined by nearly 2,000 other people across 6 continents who will spend this week learning about the progress of the Earth Biogenome Project and the very latest advances in biodiversity genomics. I'd like to also thank for the support of all the folks at the Wellcome Sanger Institute. This virtual conference is open and free to all everywhere in the world. In particular, I'd like to give a specific shout out to Dr. Kirsten Howe, who has been the leader of the organizing group for this conference and has done absolutely magnificent work with her group as the lead organizer. Now, today I will review some of the progress of the Earth Biogenome Project, and I'll present for the first time publicly the goals for the project based on a 3-day workshop held in Lausanne, Switzerland in May of 2023 that was sponsored by the Frontiers Publishing Group. Now, as most of you know, the Earth Biogenome Project has the well-publicized goal of sequencing 1.8 million named species of eukaryotes in a 10-year period. To accomplish this, an international network of networks has been established that creates and disseminates the standards for sequencing, annotation, informatics, analysis, sample collection and processing, the entire pipeline of activities across the Earth Biogenome Project. And it coordinates all of the activities of the Earth Biogenome Project affiliated projects across the entire world. There are 58 of these projects that are based in 18 countries with probably over 2,000 participants. We have a hard time counting them all. And the Global Coordinating Secretariat for this entire effort is located at Arizona State University with the support from the University of British Columbia and the Wellcome Sanger Institute. Now, these projects, these 58 affiliated projects, are distributed across 6 continents, with the largest number of projects being housed in North America, Europe, and Asia. It's very clear that we need to have more representation from the Global South. We have one project which is based in Africa, the Africa Biogenome Project, but that project involves more than 22 countries. So if you look at it on the basis of where these 58 projects are actually being conducted, it's more than half of the countries of the world. But clearly we need to have more involvement from the world's megadiverse countries and biodiverse hotspots, and we are certainly working on that. Now, the This slide shows a summary produced using the Genomes on a Tree system, which was created by the folks at the Wellcome Sanger Institute. As of this week, let me just move this on my screen here. As of this week, the Earth Biogenome Project has blown past 3,000 genomes, 3,000 species. And that puts us about a third of the way toward our revised goal for EBP phase 1 of 10,000 genomes that I will talk about a little bit later in my presentation. These species, these 3,000 species, represent about 0.2% of the total number of named eukaryotic species, about 2% of the genera, about 11% of the families, about 23% of the orders, about 25.5% of the classes, and about 37% of all the eukaryotic phyla. And I'll add a caveat here that these estimates are underestimates because at this time, not all of the 58 affiliated projects have been integrated in the GOAT system. In fact, the number as shown in the upper part of the slide is 43 out of 58. And if you look at the distribution of species sequenced by phylum, you can see that we have a preponderance of insects, of arthropods, and insects, which is maybe not to be, which is not unexpected, given that up to 80% of all the named eukaryotic species are in fact insects. After the insects, we have a good representation of chordates and of plants, still coming up, less than 300 species of, about 200 species of plants, and a smattering of other eukaryotic phyla. Important to note here that we have very few, if any, single-cell eukaryotes, and it's a very important and challenging area for the Earth Biogenome Project to develop methods for sequencing, doing high-quality reference genomes from these organisms, because they represent a significant fraction of the total diversity, eukaryotic diversity on Earth. If you look at the productivity of the Earth Biogenome Project affiliated projects, you can see that we have nearly doubled on the left side the number of species in the last 2 years. The dark blue color bar shows chromosome scale assemblies. So the fastest growth has been in the number of chromosome scale assemblies and scaffold assemblies. And there are really very few, less than about 500 assemblies in the contig space. I think this is really great, and it shows all of the efforts put in by the Earth Biogenome Project team of investigators and the leaders of the affiliated projects at creating standards and then be able to execute on standards for sequencing and assembly. The number of families is actually growing, but you can see it's sort of planing off here a little bit. There's, as I mentioned, maybe 10% of the families, about 1,000, a representative species for about 1,000 of the families has been completed. And this represents actually the reality of how the project is being conducted, as many times species are selected not for phylogenetic breadth, but for their importance to conservation, importance to agriculture, or other factors, which we will discuss also in a few moments. The thing I think that is most impressive that we have about the Earth Biogenome Project today is the fraction of the reference quality genomes that have been produced by EBP affiliated projects versus the number of reference quality genomes produced by the rest of the world. if you look here on the left side at the species level, as of this week, more than half of all of the reference quality genome assemblies, there are about 3,764 that meet the Earth Biogenome Project criteria for reference quality genome. More than half of those have been produced by the EBP affiliated projects. And it gets more impressive as you move to the genus level, nearly 60%, and now over 68% of assemblies reaching the EBP metrics for individual species have been produced by EBP affiliated projects. So my hat's off to the leaders of all of these projects who are producing reference quality genomes, as the EBP is now clearly leading the world by a wide margin in the production of reference quality genomes. And I'll again emphasize that not all projects have yet been integrated into the GOAT system. And where are these projects being, assemblies being produced? And as mentioned, there are 48 projects that are now reporting, but we have, a couple of projects that have really been leading the way for the Earth Biogenome Project. And I want to make a shout out to two of the leading projects, the Tree of Life program at the Wellcome Sanger Institute, which includes the Darwin Tree of Life, the Psyche program that you see there, and Aquatic Symbiosis project, and other projects as well. Together, the Darwin Tree of Life has produced about 2/3 of all of the genomes produced by the EBP to date, more than 2,000 assemblies. And another shout out, of course, to the Vertebrate Genomes Project led by Eric Jarvis at Rockefeller University for the amazing work over the last 7 years in leading the way and establishing the standards for sequencing and assembly. It's just fantastic to see this progress being made throughout the world. There are other projects, as you can see, the numbers are not trivial. For example, the California Conservation Genomics Project with their assemblies, and others in the hundreds of assemblies that are now have been completed. And we expect many of these projects now to come online in the next few years, which will lead to a rapid increase in the number of assemblies being produced by EBP affiliated projects. Another way to look at the data, which normalizes it instead of looking at species, is looking at the amount of sequence produced. And as you can see, like the Vertebrate Genome Project has focused on larger genomes, and so this gets somewhat normalized by genome size. But still, the numbers here in terms of bases sequenced is really, really impressive. Altogether, well over 3 terabases of DNA sequence being produced by the project, by the affiliated projects. I'm going to switch gears now and talk to you about what I mentioned earlier, and that is the results of a workshop that was held in May of 2023, and for which a manuscript has now been produced and submitted for publication to Frontiers in Science. And this conference focused on the blockers and solutions for scaling the production of reference quality genomes. I know this is a busy slide, so please bear with me, but this is our revised strategy for the Earth Biogenome Project Phase 1 and Phase 2, revealed, I guess, publicly for the first time. Phase 1 will still be sequencing approximately 10,000 species. We had 9,600 species before, because the original goal for Phase 1 was a representative species for each of the eukaryotic families. The revised goal is to have a representative of about half of the eukaryotic species in Phase 1, and completing the rest of the eukaryotic families throughout Phase 2 and Phase 3. So these are overlapping phases. Phase 2 will involve the collection of 300,000 species and the sequencing of 150,000 unique species representing approximately 50% of the eukaryotic genera, with the rest of them chosen for their importance to conservation, ecological or societal impact, or cultural significance. So we have three pillars for the project: adaptive sampling, which I just discussed. The second is producing genomes of the highest quality. The original plan for EBP was that phase 1 would produce reference quality genomes, and phases 2 and 3 would be draft quality genomes. And the primary driver for this was costs. But costs have been reduced significantly. And we now feel, the leadership of the project now feels and has done a very rigorous analysis to show that we can produce these 150,000 genomes that meet the EBP quality metrics, all reference quality genomes for these 150,000 species for phase 2 rather than draft sequence. And also that is going to be true for phase 3, and we can do this all at a cost that's below the original cost estimate of $4.7 billion over 10 years for the entire project. And new methods and new standards are being developed for myobiota and microbiota, which will allow the previously inaccessible genera and species to have their genome sequenced to a reasonably high quality standard. And we're going to do all this through global leadership, our third pillar, and equitable partnerships for phases 1 through 2 and 3, enhancing global coordination. By enhancing global coordination, building sequencing capacity across the world, democratizing genomic skills, for example, through the Biodiversity Genomics Academy that occurred last week that had more than 6,000 participants from around the world, and doing it with justice, equity, diversity, and inclusion, and access and benefit sharing across the EBP for all peoples and all countries across the world. So this is no trivial matter, and I'll wrap this up in a few minutes here. We need to scale to get to where we need to go from where we are today. And actually, to complete phase 1, which is going to require about 7,000 additional species to reach the goal of 10,000. We already have 3,000 done, and it's still October, so we'll probably be close to 3,200, 3,300. We need to sequence about 3,500 species per year globally over 2 years to complete phase 1 by 2026, and this is certainly within our range. And this will require, from here on out, to reach that goal of 3,500 species a year, 67 reference quality genomes per week. And right now, if you look at the current production, we're about 20 genomes per week across the world for all projects. So we're going to have to scale up about a 3.5-fold from the current phase 1 output. And we think with the current improvements in technology and all these new projects coming online, that this is completely within reason. And this will be done at a revised phase 1 cost of $265 million. That's $375 million less than the 2018 estimate, again, due to advances in technology, the improvements really in simplicity, and the simplicity of producing these reference genomes. Instead of 5 methods, which were used a few years ago, we can now do it with 2 or sometimes even just long reads. And this is at an average fully loaded cost of about 26,500 genomes, as described in our submitted paper. But the picture is particularly challenging for phase 2. To get to 150,000 reference genomes in a 4-year time frame, we need to do 30,500 species per year globally over the next 4 years. That's 721 genomes per week, and that will require an 11-fold increase in phase 1 output. And in our paper, we talk about solutions to this scaling problem, and Mark is going to address some of those issues in his talk. And all of this can be done at a cost of just over $1 billion, 1.1 billion, which again is $756 million less than the original estimate we made in 2018, as presented in the manifesto published in Proceedings of National U.S. Proceedings of National Academy of Sciences. And again, all of these can be done at reference quality at a projected cost of $6,100 fully loaded cost per genome, and that reduced cost due to technology improvements and economies of scale. So the challenges and blockers that we debated over those 3 days and discussed and came up with some solutions, potential solutions looking forward, involve around these 6 areas. The coordinated sampling of 300,000 species, how that would be done, solutions for scaling the coordination of species involving more people throughout the world, bioblitzes, and various strategies. But that is a significant challenge to get 300 taxonomically identified and vouchered specimens for DNA sequencing. The entire pipeline of from sample to sequence to assembly was discussed, and solutions for that. Scaling issues were presented. High quality annotation. Annotation is a particular blocker right now of 150,000 genomes, but new developments in making annotation software available to all of the EBP affiliated projects will help us to release the backlog of genomes that we have right now even, and this is absolutely necessary. Innovation in this pipeline is absolutely necessary for us to achieve that goal. How we deliver impactful analysis, for example, how do you scale alignments for 150,000 or eventually more over 1 million species? Integrating innovative and planet-friendly informatics. Of course, computation is energy intensive, and how are we going to deal with those issues? And importantly, how do we scale the global biodiversity workforce to be diverse and inclusive and active throughout the world so that we can achieve these goals? And the exciting thing, I think, and that we're beginning to hear about, is how these genomes then are being now used in ways which are leading to incredible discoveries. These are just a few examples over the past few years of EBP affiliated projects. There are many groups involved in now producing papers from the Vertebrate Genomes Project assemblies, and the genomes being released by the Wellcome Sanger Institute are open to anyone to participate in analysis immediately with no restrictions. And so we're entering a very, very exciting phase of the project where the science being done with these incredibly high quality genomes and the efficacy and the utility and the strength of these reference quality genomes are being demonstrated in ways in which even we could not have foreseen 8 years ago when we began on this long journey. So I want to thank everybody very much for their attention, and I'm going to turn it over to my colleague and friend at the Wellcome Sanger Institute, Mark Blaxter. Mark?
What I'm going to do is take what Harris has presented, which seems a really challenging prospect to really get this crazy project going, and show how we at the Sanger Institute and the Tree of Life have been pushing for this to happen, how it's demonstrated, I think, that we can do it. And I hope that's what people take from my talk, is that it is possible, and that dream of sequencing everything is possible. And also talk a little bit about how we might share those approaches that we're developing globally. So So I've retitled this talk, Sequence Locally, Think Globally, because that's what I hope we're doing. We're using our local experience at the Tree of Life to try and think about how we can make this a global effort. So in astronomy, they have some wonderful tools. Here's the deep space view from the Hubble telescope. They pointed the telescope, astronomers pointed the telescope at the dark space between the stars, and lo and behold, they found thousands of galaxies, each of them containing hundreds and hundreds of thousands of stars. So the new genomics technologies we have, I think, are a bit like our Hubble telescope or the Kepler telescope, in that they allow us to see much further and get much, much more detail about the living world around us. And I think these new tools we have, the long reads, the long range technologies, the new assembly technologies, actually allow us to see things we've never seen before. So I know it's a bit like, I'm a genomicist, I do genomics, I look at a species, I look at you, and I think that looks like a nice genome. But really, I think that sequencing all of biodiversity is now within reach, and we have the tools to do that. So at the Sanger Institute, we're really privileged to collaborate with an amazing group of people. We're not really a biodiversity institute, we're a genomics institute. And so we've collaborated with many organizations who are, if you like, real biodiversity institutes, people who can identify and collect for us and with us, and who help us prioritize what we're sequencing. So we're involved in a number of projects, from our flagship project, which is the Darwin Tree of Life project, sequencing all the eukaryotes that we share these little islands off the west coast of Europe with, so the islands that are home to the nations of Britain and Ireland. So that's been our major project. We're also collaborating more widely from a new project called Aegis, which I'll introduce briefly in a minute, the Aquatic Symbiosis projects, Project Psyche, which is sequencing Lepidoptera, and of course with the Vertebrate Genomes Project, the European Reference Genome Atlas, and the EBP. So we're really excited at the Sanger this week because we have hit a milestone. So it's not just 3,000 for EBP. We've hit 2,000 genomes produced by the Tree of Life project since we started. This number has been a long time coming. We've watched it approach, but it's really exciting to finally see it. So this is a graphic which will appear on a t-shirt near you soon, which is one little square for every species that we've sequenced, and each little icon in the squares, which come from Philopic, is indicating of what sort of species it is. So we've sequenced from everything from single-cell protozoa to very large trees, to mammals and birds, fungi, and many, many plants. I'm just going to illustrate some of these quickly, just because it is so exciting to get to this number of 2,000. So the 2000th genome that was made public was of this very interesting little plant. This is alternate water milfoil. It's a plant that lives in ponds and in slow running waters. So it's a plant that's gone back to the sea, to living in water. It's a major component of many freshwater ecosystems. It does a lot of cleaning of the water and of stabilizing sediments. But interestingly, it's also an invasive plant. It's very good at growing in freshwater. It's called a pernicious weed by the North American EPA. So this plant is of use, understanding its biology, but it's also, the genome may also be in use in controlling this plant where it's growing, where it's not wanted. Again, this genome, it's a small genome, 245 megabases, but it was actually quite difficult to assemble. So we're actually quite proud of this one. This is the 2000th one that made public. We actually submit our genomes in batches. So it's really hard to say what was the 2000th one we actually released. This is the 2000th one that appeared on the databases. So I've got a few other milestones that we passed at approximately the same time as passing 2000. One is that we passed our 1,500th sequence for the Darwin Tree of Life project. That was this is a beautiful caterpillar, if you like, caterpillars. The puss moth, a large moth native to Europe. And so this genome was the 1,500th genome coming out of Darwin Tree of Life. We're hoping to sequence something like 8,000 Darwin Tree of Life species in the next, to get to 8,000 Darwin Tree of Life species in the next few years. This is with collaborators across the islands. We also submitted our 150th genome for the Aquatic Symbiosis Genomics project. This is a project where we're trying to sequence the host and also the microbes, eukaryotic and prokaryotic, that live with them. It so happens that 150th was this amazing snail which lives on deep sea vents in the deepest of the oceans, and it is called the scaly-footed snail. It has these strange scales on its foot. So this is like an ordinary snail with these strange scales on its foot, and these scales actually incorporate metal. So they're maybe part of sequestration of toxic metals from the organism, but these organisms also live in symbiosis with hydrogen sulfide fixing cobionts or symbionts. So that's our 150th Aquatic Symbiosis Genomics genome. Project Psyche is a new project. We started sequencing the Lepidoptera of Europe. We're collaborating with people across Europe to collect butterflies and moths. There are about 11,000 of them across Europe. And this has been an amazing project to kick off because we had a great collecting season this summer, collecting many, many, many species. And while Darwin Tree of Life has also sequenced butterflies and moths, for Project Psyche, our 100th genome approximately was this dingy skipper or Mediterranean skipper butterfly. It's called Nostradamus. Maybe that's telling us something. Nostradamus was a famous seer who looked into the future, and apparently you can predict what the future is going to be like. And what we hope is that these butterfly and moth genomes are going to be very useful, for example, for looking at the effects and impacts of climate change across the continent. We work very closely with the Vertebrate Genomes Project. This is just a very pretty one, and we've curated many of the genomes from the Vertebrate Genomes Project. This is number 187 of the genomes that we've sequenced and released. The red-billed tropic bird. It's found across the tropics, very interesting distribution. So it's found in Caribbean and the tropical Atlantic, but it's also found in the Indian Ocean and in the Western Pacific. So a globally distributed bird. It's probably not individually as a species endangered, but it will probably be a very good indicator of the health of tropical ocean ecosystems. We work with the European Reference Genome Atlas. This is an amazing distributed network across Europe, trying to bring many, many nations and institutions that don't traditionally do genomics into the genomics field. This was the 22nd we sequenced. It was part of the bunch that was released around 2000. This is a lichen, a beautiful symbiosis between an alga and a fungus. And again, we can sequence both those genomes. So really, this is number 22 and 23, because we get 2 genomes for the price of 1 for this one. Our newest project is AEGIS, which is a wonderful acronym that stands for Ancient Environmental Genomics Initiative for Sustainability. Yes, we crammed the name into the acronym. The idea of this project is to use ancient DNA from sediments of lakes and agricultural and other sedimentary ecosystems across the world to understand what the genotypes, what alleles were present and circulating in species as the planet has gone through previous climate change. So it's led from Copenhagen, the group of Eske Willerslev. We're a reference genome sequencing partner in the UK, and we're sequencing reference genomes for plants that we expect to find material for in the sediments, such as the milfoil, which was the first genome I presented, but also this one here. This is the first official AEGIS genome that's come out. It's of Aegilops tarshii, which is the one of the ancestors of modern wheat. This species contributed the D genome. So we sequenced this in collaboration with the National Institute of Agricultural Botany in the UK. Again, beautiful genome. This high C plot shows the 7 chromosomes, and the scaffold N50 here is 563 megabases. We're also trying to sequence the very small. We have a project to sequence as many nematodes as possible. Those of you who know me know that I think nematodes are the best thing on the planet. So Erna King and others are working to sequence as many nematodes as possible. And to do this, we've had to develop new methods for getting good genomes out of single specimens. So this is Odontocalamus thalassophagus, a predatory nematode of marine muds. And this genome assembled by Erna comes from the PacBio data come from 1 nematode. And the high C data came from 22 individuals of the same species, and it's assembled into 14 chromosomes. So we now can approach using PacBio and the method that we use here called PIMS to sequence things down to the size of myofauna. Not quite to single cells, but we're getting there. Just to emphasize that when we sequence things, as I said, with the symbiont systems, for example, the lichen, we're pulling out 2 genomes. In the lichen, it's an alga and a fungus. What we also find in many species is that they're infected with other things. Now, 33% of all eukaryotic species are classifiable as parasites. 33%. So a third of all the species we're after ultimately are parasitic on something else. And so we're developing methods to pull out those parasitic genomes. This is work by Amjad Khalif in the Sanger Institute, who looked in the genome of this wasp, Sphecoides crassus, and noted that there is a set of contigs in the primary assembly which actually map to microsporidia. So microsporidia are relatives of fungus. They've got very, very small genomes, but they are eukaryotes. So he has managed to assemble over 30 microsporidian genomes from various species we've sequenced. So this is, if you like, the bycatch, the cobionts, the other organisms that are there. This is especially true, the sequencing other things is especially true, obviously, in our aquatic symbiosis project, where we're deliberately trying to sequence both host and cobiome. And this is a pretty graph. I'm not expecting to get much biology from this. The tree in the center is of the bacteria we've sequenced. So from about 780 different bacterial species, we've managed to assemble metagenome assemblable genomes from these. So about 5,500 genomes we've assembled from the Aquatic Symbiosis Genomics Project. And these, the rings around the outside are showing the presence of these bacterial taxa in the various groups of organisms we've sequenced. So these range from mollusks and annelids through jellyfish and other protists. So we have a huge wealth of bacterial genomes and archaeal genomes and parasite genomes coming on for free along with our host genomes. We're trying to get these data out as fast as possible. And for many years, we've been, 5 years, we've been publishing them in genome notes in Wellcome Open Research. So this is, genome notes are a very short publication that say, we've sequenced this genome. This is how good it is. I mean, it really is a good genome. Please use it. And we just submitted our 1000th genome note. So the 1000th genome note was actually from the whiskered bat. And this is a project which is both Darwin Tree of Life and BAT1K. And so this is an announcement that the genome is, as Harris said, available for use and available for reuse everywhere. We're very lucky in the Sanger Institute that we have a very close relationship, obviously, with Wellcome Open Research. We are Wellcome funded. And publishing genomes is an expensive business if you go through the traditional route. And so we've been working very closely with Earth Biogenome Project and the Knowledge Foundation to think about how we could make a genome note platform that could be accessible for everybody. So we're sequencing locally and using local resources, but how can we make this available to everyone? So this publishing method is not available to everyone, and it is expensive. It can cost as much as sequencing the genome has. So we've been working on something we call the universal genome note. And so the idea is to build a repository of genome notes, which is open, which is accessible for all, but which is also very, very cheap to publish in, that will mint a digital object identifier for every publication, will be very interactive and cross-referenced, and will be customizable for different projects inside EBP. So those of you interested in this will be presented at the Earth Biogenome Project membership meeting, if you like, on Friday. So do come along to that, and Knowledge Futures, who built this, will present the idea to the community. So I'm very aware that we are meeting at the same time as COP 16 in Colombia. And COP 16 is talking about digital sequence information and about access and benefit sharings, especially for the biodiverse nations of the world. And I'm very aware that we live in a time of extreme crisis. This is the NHM Biodiversity Intactness Index, published last year, where the darker colors indicate intactness, and the colors that look like the place is on fire is where the biodiversity is not intact, is very disrupted. And we're fighting a battle against our own depredation of biodiversity. So we want to see if we can get biodiversity genomics out to the places where biodiversity is still intact, and also biodiversity out to areas where biodiversity is under threat, so that 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 genomics can be used to aid restoration and conservation of biodiversity. To do that, we've had an idea, it's not a new idea, of putting a genomics laboratory in a box. This G-box idea is that we would supply a fully equipped genomics laboratory, which would come with guaranteed training and support from established genomic centers, and also guaranteed funding from, guaranteed support from the technology providers and the reagent suppliers, such that wherever this was put, whether it's in Guyana or the Bahamas or Kenya or Malaysia, it would get support from the companies who deliver to our doorstep on 24-hour notice. We need to have that for there as well. So the idea is to build a package where we can support the delivery of genomes locally. So we think one of these boxes can deliver between 1,000 and 2,000 genomes per year once it's up and running. So having 25 or 30 of these around the world would allow us to meet phase 2 goals. I've got a video, but I think we're out of time, so I'll skip past that. Sorry, if you want to see the video, contact me. I'll show you a video. We have a fly-through, a lovely fly-through built by marshals who've collaborated with us in building this. Stop it running. So what I hope is that in the near future, we got these beautiful 2,000 genomes from Tree of Life. We're going to generate, we hope, 2,000 in the next year, or additional genomes the next year or so, but other sites will be springing up that can deliver to exactly the same specifications and at equal rates. And that way, we're going to meet these extended goals of EBP phase 2 and really sequence life for the future of life. Thanks very much.
Wonderful. Thank you, Mark and Harris, for sharing broad overviews and really specific overviews. To our audience joining us, please drop your questions. We have a few moments for those. We actually already have one, which is about, the specific question is how small labs can join the Earth Biogenome Project, and about accessibility to tools, whether or not you're an affiliate. And so I'll raise that actually to both of you, both Harris as sort of considering the large entity of EBP, and Mark, you as well, from some of the specific tools coming out of your project.
Yeah, I'll start. You know, it does depend how you define small. If small is between you know, 1 genome and 15 genomes, say, we recommend in the future that these projects become allied to the regional node system that is being developed within the EBP. And regional nodes will be continental or you know, in a particular geographical region, subcontinental. So if you have a project that's that small, you know, please contact us, and we will plug you in to the network of because this is a network, international network of networks. We will plug you into a network that will allow you to participate directly in the project. If your project is, you know, on the order of 100 genomes, you should consider joining the project as an affiliated project. Given the scale of the way things are moving along now, 100 genomes is still a modest, if not small, project. This is the way we're thinking now. So if you've got funding and you've got all the resources and samples and so on to do 100 genomes, please consider joining as an affiliated project, and the instructions to do that are on the Earth Biogenome Project website.
Sure, I can answer as well. So in Europe, the European Reference Genome Atlas community is explicitly set up to encourage and develop just that idea of having small labs who are producing genomes and accessing the tools and the expertise of people who've been doing it for maybe decades, and also people who are having new ideas and new ways around, because everybody has new ideas which work, and it's exciting to have them. So here at the Sanger Institute, we try and be as open as we can with our protocols. So we have a dedicated protocols.io website, which has all our protocols on it. I can show that just quickly, protocols.io. So this is protocols.io, and we've put on protocols.io all the protocols that we use in our lab to generate our genomes. This is, if you like, from the sample to the DNA being submitted to the sequencer. And then for the analysis part, we publish all our pipelines that we use. For example, the tools used for curation or for cleaning the genomes to remove contaminants are all on our Tree of Life Pipelines website. And again, you can download the Nextflow pipeline. So I think one of the ways that projects such as the one I'm part of can really help others is by making our tools public and useful, therefore. But I think we also have to be very open to people saying, actually, this doesn't work for me. And so I would very much hope it's a conversation that people will install our tools and say, how does this work? And so the third bit of it all is the Biodiversity Genomics Academy, which is just finished. And Harris mentioned the 6,000-7,000 people that took part in that. The idea there is that we get people who have written pipelines or who are the software developers who built tools or have special approaches to doing things, whether it's in databasing or anything else, can show what it is that they do and can run a masterclass on being a genome biologist. So I think we're publishing things openly. We're making our tools open as we can, and we're trying to promote things like the Biodiversity Genomics Academy as a way of people getting up-to-date training in the tools that we generate.
Yeah, thank you for sharing those, Mark. I mean, it's really great that these tools and the academy is happening. And you mentioned a word that I just want to touch on briefly in the couple minutes we have left, conversation. And so the academy is sort of at a specific time. The tools assume that you maybe have some level of understanding. I'm wondering if you both can touch on how can people engage in this conversation? So a bit just more practical, if you're just starting out, if you have more complicated sort of getting off the ground running. You touched on GBox, but maybe even before that, how do people enter into this conversation to join the work that we're doing?
I would say contact us. And we have a steady stream of visitors at the Sanger Institute who come through to look at what it is we do. And especially we get people coming in wanting to learn specific parts of the pipeline. And so that's what I would say, contact people. I think that the biodiversity genomics community is exceptionally open in terms of science communities. This was, if you like, historical because of the way the Public Human Genome Project was set up as an open project. And also the way that, for example, the Vertebrate Genomes Project has had an open Zoom call every Friday for as long as anybody can remember. And everybody is very welcome to join that and you know, say what their problems and questions are. Sometimes the meetings will be way beyond where you are, and sometimes they'll just be absolutely on the nail. But again, that brings you into the community. The community really does exist.
Yeah, I'll add, in addition to that, that we have a very well-established committee structure in the Earth Biogenome Project, which has an LC committee. It's not just the technical focus of the project. There are committees that deal with ethical, legal, and social issues, as well as justice, equity, diversity, and inclusion, as well as the scientific committees. And if you are interested in engaging in conversations, please go to the website, contact those chairs. There are occasionally, every few months or during the year, considerations for new members of these committees. We particularly welcome the participation of younger people into the conversation, Sadie. And the committee structures are a great way to engage. If you have general inquiries about participation, the Earth Biogenome Project Secretariat has an open line of communication. There's contact information there on how to contact me or Nicolette Caparello, who's the Director of Communications, as well as other folks on the EBP Secretariat staff who can answer your questions and direct you to different groups within the EBP to which you may begin your engagement. The final thing I will say is the EBP is considering ways to bring individual members. Right now, EBP is a network of projects, and we need to find a way to have a broader engagement with the thousands of individuals around the world who are interested in biodiversity genomics, and we will be discussing that issue in the coming year. Thanks for that question.
No, wonderful. Thank you, Harris, for giving us a broad overview of where the EBP is and where we're going by building global leadership through equitable partnerships. And Mark, thank you for illuminating a specific way that we might get there and sharing all the beautiful species of how we might make this a reality. I want to remind you that we have many exciting sessions ahead this week, including 2 keynote sessions later this afternoon. Anne McGuy from the African Biogenome Project, and David Shani from the Genomics Aotearoa Project. I also want to remind you of dedicated sessions for early career researchers, ethical, legal, and social issues, justice, equity, diversity, and inclusion, and indigenous data sovereignty. Vastera, continuing the tradition of this conference, is going to be our art and science contribution. You can join them at various times throughout the week. Vastera is brought to you by Lani, Hector, Rafael, Diana, and Daniel. In August 2019, they were formed in collaboration with the United Nations Museum as an audio platform with the purpose of raising a voice of awareness about the care of our natural ecosystems. And lastly, I want to thank many, many individuals that have brought this to you: the Scientific Planning Committee, our sponsor, the Tree of Life, as well as our AV and Tech team, which, true to their name, they want to be behind the scenes. I tried very hard to get their name and they refused to share, but they are many individuals that have contributed to the success of this project and this meeting. And lastly, like Nostradamus and the myth of being able to predict the future, I wish you a wonderful Biodiversity Genomics 2024 as we continue to endeavor together to create the world we want of living in harmony with nature. Thank you and enjoy the conference.